do the combining to, or I don't know whether we can develop even more than 2 molecules into one big consortium. It's interesting because normally we formulate several micros, but this time you are formulating several molecules or 2 molecules to get together and then work as an antimicrobial. And I hope that not only under microbial activity, we can actually develop or customize, yeah, the purpose of application, not only in health, maybe we can also later on develop in agriculture, for example, environment, and yeah, many purposes. But the key technology, you are the expert. Yeah. And then I hope that participants here, in especially in this room, today, I hope we can have like a fruitful discussion, effective discussion, and then from this webinar, we hope that we can develop kind of collaboration hosted by Dr. Aya. I think will be the role model, the first collaboration between and Monash University, I hope. Yeah, and then let's make it this year or soon, yeah, in the future. I know that we had initiated a couple of years ago, maybe a bit of it, but there is some, many challenges, and I hope we can overcome this time. So I'm looking forward to hearing the good news, signing the project agreement between BRIN and Monash University, specifically on a small molecule and hybrid compound. This is really interesting, really. And then I would like to inform you, everyone here, that we BRIN are going to develop. We have a collaboration platform, structural biology, as you may have been informed. And then, yeah, this year we focus on micromolecule, and then this year we are going to develop also on micromolecule, which is small molecule will be included. Yeah, and then very interesting, and then I'm sure that we can, make a deal in one project, specific project, and then yeah, submit proposal how we can fund this small using small grant, or yeah, it depends on the scheme. BRIN has several schemes, as we discussed yesterday. Yeah? I hope that once we have collaboration, good collaboration, we can apply the degree-based program, sending our young students from Indonesia to Monash University, and also we'd like to invite you and your team as a visiting researcher working together with our young researchers here. Dr. Anya is the lead for this kind of project. Yeah. So I will always monitor her. Yeah. So the progress. Yeah. How the collaboration discussion going on. And I hope, yeah, sometimes it takes, I know it takes time for the collaboration, especially internationally. So, but okay, while we are preparing the legal thing, like a project agreement, I hope this kind of joint research, can do parallel here. So, everyone, thank you very much again for your participants participation today, and to our speakers. Hopefully, we can gain a lot of new knowledge and new insights on small molecule and hybrid compound. I like the term of hybrid compound and synthetic, actually, synthetic or artificial compound. Yeah, very interesting. And I hope this project can also be supported by our platform when we once we develop the structural biology and especially microbiome. Yeah. Okay, everyone. So please enjoy the webinar and be active for discussion. Here also I officially opened the webinar this morning. Yeah, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Ahmad Fathoni, for the encouraging remarks. Before we start the agenda, let us capture this moment with a group photo. So for on-site participants, please gather at the front of the room, the center. And for online participants, please turn on your camera so we can include you in the virtual group photo. So we can invite committee to take a group photo. Dr. Anggia Prasetyo Putri is a molecular microbiologist and group leader at the Research Center for Applied Microbiology, BRIN, Indonesia, with a Ph.D. from the University of Queensland and an MSc from the University of Melbourne. Her expertise lies in antimicrobial resistance, microbial biogenomics, and drug discovery. She has led and contributed to numerous international and national research projects, particularly in antibiotic development and SARS-CoV-2 surveillance, a recipient of several prestigious awards, including the L'Oreal UNESCO for Women in Science Fellowship and APEC Australia Women in Research Fellowship. She is also an active scientific author and patent holder, and mentors graduate students in microbiology and infectious disease research. So without any further ado, to Dr. Anggia, we kindly invite you to lead the session.
Good morning, everyone. Is my voice all good? Okay, thank you. Okay, good morning, everyone, and thank you for being here this morning. My name is Anggia, and I'll be your moderator for the webinar session today. So today we are lucky to have two wonderful speakers, my wonderful friends and colleagues from Monash University, Malaysia, Dr. Chee Wei Ang and Dr. Ken. And now we are going to have Dr. Chee presenting her presentation first, and then we'll have Dr. Ken later, and we'll have question and answer session after every presentation, and then we'll have a common discussion at the end. But yes, so I personally know both of the speakers, and so I can testify that they are quite good experts at what they do in synthetic chemistry and medicinal chemistry. And I think we are quite fortunate because I don't think there's that many people in BRIN who have that expertise. And I think that today is a good opportunity for us to see what is possible in terms of research projects, research topics that might be feasible to collaborate with Dr. Chee and Dr. Ken. So without further ado, I would like to introduce Dr. Chee as our first speaker. First of all, I would like to read a little bit about her CV. Apologies. Sorry, it disappeared on me. Oh, there he, okay, thank you. So Dr. Chee Wei Ang is currently a lecturer at the School of Science, Monash University, Malaysia. And prior to that, she's a postdoctoral research fellow at the School of Science. And she's also had experience as a visiting fellow for the Marie Curie Action Staff Exchanges. And she completed her PhD in medicinal chemistry at the University of Queensland. That's when I first knew her, and we became good friends since then. And prior to that, she did her MSc from University Science Malaysia and BSc also in University Science Malaysia. And she is quite a decorated researcher, having won several awards and travel grants. And also principal investigator of a number of grants as well. And she's won a number of posters and oral presentation prizes, and she has published in extensive journals with highly reputable journals. And so I think her talk on hybrid compounds today would be quite interesting to listen to, and I'm sure there will be some aspects of the research and the things that she's presenting that would be quite useful for us as the participants today. So without further ado, Dr. Chee, I would like to let you to your presentation, and time is yours. Thank you. You have maybe roughly about 45 minutes or so for your presentation.
Yeah, thank you so much. Let me share my screen first. Is that all good? Yep. All right. Good morning, everyone. Salam sejahtera. My name is Chee. As mentioned by Angya, I knew her for I think down here is close to 8 to 9 years. It's my great pleasure to be here at BRIN. This is my first time here. I had a wonderful lab tour yesterday, visiting a lot of researchers and observed a lot of the research activities ongoing here. Could you hear me well at the moment? Okay. Oh, I'm sharing the screen. Did I'm sharing the screen? Sorry.
Is that better?
Is that? Oh, okay. Great. Thank you.
No.
Sorry for the technical issues. It's always happened during the hybrid session. Okay, so today I'm just going to share with you a very brief project that was recently ongoing in my group, where we are working on hybrid-inspired compounds against bacterial and parasitic infections. But before I start with that particular project, I would just like to give an overview of what my group is working on. So perhaps if you are interested, perhaps we can talk about further collaboration in this space. So I am a medicinal chemist. So my group is mostly working on developing new therapeutic agents, particularly focusing on tropical diseases, especially those bacterial and parasitic infections. I'm particularly interested in synthetic small molecules, but I'm also exploring on natural product analogs, where I'm interested to explore on how structure modification could change the activity of the natural product parent compound. Besides that, I'm also working with Mahidol University and York University at UK to explore on metallotherapeutics, where we complex the metal ions with drug molecules, and hopefully we can use this approach to basically break the resistance issue for malaria diseases. And of course, we are not only looking into active compounds. As a medicinal chemist, we're also looking into drugs with good drug-like properties, with good pharmacokinetic profile. So what I'm working on is we use an in vitro model in my group that we use LC-MS-MS as a quantification method, where we determine whether the heat molecules or the lead molecules that we synthesize, whether they have a good properties to be proceeded. Because this is very important for any clinical candidates to be successful. They need to have a good pharmacokinetic and a good drug metabolism profile. And lastly, my group is also working on a protein drug conjugate. So this protein drug conjugate is basically used as a carrier. So a lot of cases we found that the drug molecules, they have issue to reach the site of action. So here in this case, we use a protein as a carrier, where we conjugate the protein together with the drug molecule, and we use that protein to basically bring the drug into the site of action. So this is what we call the receptor-mediated delivery system, where we use that to release the drug in a particular site. This can help to reduce the off-site effect. So all of my research interest here is basically focusing on addressing the challenges of AMR. I think BRIN is also one of the frontier that work on AMR issue. So antimicrobial resistance is not only the issue of the tropical regions, it's not only the issue for Africa countries and Asia, but it's also a global issue. So we believe that if there is no further action that has been taken to address this issue by now, we will be facing a death rate of close to 10 million in 2050, and this will also cause a great economic burden of close to 100 trillion in less than 30 years of time. So AMR is not just the issue caused by bacterial, although many cases people talk about AMR, they talk about only bacteria, but actually AMR is also the issue caused by other microorganisms such as viruses, parasites, and fungi as well. So this is just a very nice diagram that I found in Nature Review Drug Discovery, just published last year, that showcases the differences in terms of the complexity of the microorganisms that will affect the complexity of the resistance as well. So my group is not working on antiviral. I'm more interested in parasite and bacterial. But here is just to highlight that the more complex the microorganism, the more complex will be the resistance. So for viruses, they have a limited number of targets. And the main resistance is caused by the target alteration, where sometimes the viruses, they could have an increased expression of specific genes or reduced drug binding affinity to the drug and making the drug less effective. For parasites, they are slightly more complicated than viruses. The resistance mostly caused by reflux system and also the target alteration. And for bacteria, it's even more complicated, as you can see from the diagram there. For bacteria, the resistance could caused by either the drug being pumped out from the efflux system, the drug getting degraded in the cells where they lose the effectiveness, and also the limited uptake of a particular drug as well as target alterations. So here is just a snapshot of some of the bacteria that cause a high death rate of AMR. And in my group, I'm working on those bacteria as well. So we are working on E. coli, K. pneumonia, A. baumannii, and as well as other escape pathogens. I'm also working on MTB, but because in Monash, Malaysia, we do have limited resources that we could not do the MTB by ourselves because we don't have the biosafety level 3 cabinet. So we basically collaborate with our partner in US and also in Queensland to basically did the MIC and antimicrobial study on Mycobacterium tuberculosis. And I know that MTB is one of the major issues in Indonesia as well. So if anyone is interested to work in this space in MTB, I'm really open the collaboration opportunity to work with you on this area. So in my group, I'm particularly interested to look into this approach called hybrid drugs. So what is basically hybrid? So hybrid is basically means that you will link two pharmacophore or two drug molecules together as a single molecule. So not only two, sometimes you can even link more than two. You can bring 2, 3, or even 4 drug molecules together as one. So how we design it, we can design it in 2 ways. First way, we can basically link them by a covalent bond, which is stable. So in this case, this new molecule, they will exert its bioactivity as a whole molecule. Or we can also design it differently, where we can put in a cleavable linker. So this cleavable linker, when it goes to the specific site of action, they could get cleaved off. So we call this an hybrid prodrugs. So in this case, these 2 drug components that originally linked together, when they go to the body, they could be basically separated into their individual components and exert their activity. So what's the benefit of this hybrid approach? Basically, this hybrid approach, one that can potentially exert a new mechanism pathway, so they can target novel target that is not originally targeted by the old drugs, or it has the potential to have multi-targeting approaches, meaning that they could basically exert more than one mechanism action. So this is very powerful because a lot of the resistance caused by single target mutation. So if we can target multiple targets, that could really help to reduce the resistance problem. So here are some of the examples of the hybrid antibiotics that basically has been reported in the literature. So the first one, I think, is a very common example that perhaps you use it in your lab as well. We have ciprofloxacin, we have trimethoprim, both of them good antibacterial agents. When we hybridize them together as a single molecule, they have surpassed the MIC of the individual drug itself and also give better activity than its pure mixture. So the second example is the combination where we hybridize ciprofloxacin with PBD. So these 2 molecules individually, they are good drugs against Mycobacterium tuberculosis, but the problem is the PBD itself has high cytotoxicity issue. So if we hybridize them together, they can basically help to reduce the cytotoxic effect of PBD, as well as they could be effective against the drug resistance and also the drug susceptible Mycobacterium. And the last example that I would like to show you is a phase 2 clinical candidate. This molecule that basically combining rifamycin, a natural product analog, with fluoroquinolone as a hybrid drug, it could has surpassed the activity of the pure ciprofloxacin for almost close to 550-fold activity. And the best part is it has multiple mode of action, meaning that it could target not only one enzyme, but multiple enzymes. So this has proven to give good activity against the susceptible as well as the resistant strain. So these examples really inspire us in our group. So today I'm going to share with you 2 projects that I'm working on. The first one is something that I worked since my PhD time. We have obtained some very interesting results, and that's why it inspired us to pursue this pathway into my second project, which is the quinoline-based hybrid. So for the first project that I'm going to share with you, it's basically we use the hybrid-inspired approach where we take the fragments, the active fragments for the individual drugs and fuse it into a single one, and we're able to synthesize a novel scaffold from there. So the first molecule that we take the inspiration is from pritomonide. So pritomonide is a known TB drug. It has been approved in 2019 for multi-drug resistant TB. So we took the fragment from the nitroimidazole ring, which is colored in blue there, and we also took the active benzyl substituent, which is colored in yellow, and then we fused them with another ring from benzothiazinone, PBTZ169, where we take the 6-membered ring, the one colored in green here, and we do some modifications, and we managed to synthesize this new molecule called nitroimidazole pyrazinone in just 5 steps reactions. And this is a work that we have done some time ago, has been published previously, and if you're interested, you can perhaps have a look at the synthetic pathway. So when we compare the activity between these 2 molecules, the pritomonide, the known TB drug, with our own molecule, we do see a very similar activity in terms of the Mycobacterium tuberculosis. The best part is that it is non-cytotoxic against mammalian cells up to 100 micromolar, and it's also highly selective, meaning that it is not active against the non-tubercular Mycobacterium. It is also non-active against escape pathogens, meaning that they are very, very selective to only Mycobacterium tuberculosis. And as a chemist, we like to design and play around with molecules. So we continue to synthesize a library of different derivatives. And we found that we can basically modify the side chains. And the nitro group is important, so we can't really do much on the nitro part. But how about the middle ring here, the cyclic ring here? So we are interested to see if we can still modify and make changes. So in this case, we propose to design another 3 more scaffolds. So the first one, scaffold B, is the regioisomer of the pyrazinone ring, where the substituent change from N to O substitution. And then we also designed to make it a reduced ring size. From 6-membered ring, we make it to a 5-membered ring, which is the scaffold C. And then we also invert the amide link. So in this case, we make it from scaffold D, so which has a revert amide linker. So this means that the substitution, instead of the 7th position, we make it to the 8th position. So for all these changes, unfortunately, we find that none of them retain the activity. They all have completely lost of the Mycobacterium activity, meaning that pyrazinone is the important structure. So if we need further modification, we can't touch on that particular ring, but we can do further modification on the substitution. So as I mentioned earlier on, we are not only focusing on activity, we are also focusing on the properties, because for any clinical candidate to be successful in the clinical phases and in the development stage, they need to have good properties as well. So in this case, we are looking into addressing the solubility issue. So solubility issue is always the problem for known nitroimidas or TB drug in the market. For example, delamanid, the known TB drug, it was found to have low solubility, and therefore it requires twice daily dosing. And this could be a problem to make patients to have high burden rate because they have to keep on taking drugs at least twice a day. So this can reduce the patient adherence. So in this case, we also investigate the solubility issue of our own compound. So we plot a correlation curve. So how does this correlation curve comes from? So the y-axis is basically the activity of against Mycobacterium. We plot it as PMIC value. The higher the PMIC value means that the higher the activity. And then the x-axis we plot for solubility. So solubility represented by log S value. The more negative the log S value, meaning that the lower solubility it is. So here we can see that the one circle in the pink color region is the one that gives best activity, but unfortunately they are having low solubility issue. So similar issue that we face together with the nitro drugs in the market. So therefore, as a chemist, we always wanted to do some modification in order to improve that activity. So what we did here is we try to do a few modifications to reduce the lipophilicity of the compound. So the first thing we did is we include the polar linker. Second thing is we change the benzene substituent with something less lipophilic heterocycles. And lastly, we include the extended ring system, trying to mitigate between solubility and activity at the same time. And when we get the molecule, we call this a second generation of analog. We plot them again in our correlation curve. And this time you can see that the dot there, all different colors represent different series. The one in black is basically the first generation analog that we synthesized. The one in blue is the one having the less lipophilic heterocycles. The one in green is the one with polar linker. And lastly, the one in pink color that you see means the extended ring system. So I have divided this correlation curve into 4 quadrants. The one in the the one in the light yellow quadrant is basically means that compound with good activity and good solubility is the one that we're aiming for. But unfortunately, as you can see from the correlation curve, most of the modification, when they have improved solubility to value of -3 to -5, they tend to have low activity as well, meaning that they have PMIC of lower than 5, unfortunately. So this makes us rethink that, is the lipophilicity an important parameter for a drug molecule to be successful as a clinical TB candidate? So why we have this thought is because we know that the Mycobacterium is different from other gram-positive bacterial. They have an issue where they have a very lipid-rich mycolic acid layer on top of their outer membrane. So this makes them very difficult to have the antibiotics to permeate through the cell wall. So because of that, we plot again our curve now in order to find out whether the lipophilicity plays a role in their activity. So again, this time, we plotted PMIC at the y-axis. X-axis this time replaced with log P. So log P is an index for lipophilicity. The higher the log P value means that the higher the lipophilicity of the compound. And we plot them for all the compounds that we synthesized in the library. It has close to 80 compounds in total. So what we see again is that those that have a log P less than 0, meaning that between -2 to 0, they are basically means they have low lipophilicity. They have low lipophilicity. At the same time, they also have low activity because you can see the PMIC value is low. So for those active compounds, the PMIC that is larger than 5, when we translate to MIC, it means an MIC of lower than 10 micromolar. They have the log P value range between 0.75 to 4.1. So this is kind of like a very broad range of the log P value. And when we look back again at the lower region of our plot, we do see that there are also some compound, log P higher than 4. They also seems to be not really active against MTB. So this tells us that yes, lipophilicity may be an issue, because if the log P is too low, less than 0, they are not active, but that is not the determinant factor. That is not the only factor that contribute to the activity. They could be also contribute to other factors as well. So we have temporarily closed the story of that part first, but we now move on to the efficacy. So because of the promising activity that we found in vitro, so we pursue some in vivo study. So the study was basically done in the University of Illinois by our collaborator. So he basically helped us to do the in vivo work on mouse model, where we provide the 5 days a week treatment to the mouse using our own compounds. So we selected 2 compounds here, compound 9 and 14. They have relatively acceptable oral bioavailability between 64% to 98%. And we gave the mouse, we treat them in oral treatment for 21 days. And at day 31, we sacrifice the mouse and then we calculate the bacterial count in lungs. So in the graph, as you can see below, our compound 9 and 14 basically has show a significant reduction in terms of the bacterial burden compared to the vehicle control. But when we compare with pretomanide, yes, compound 15 has a similar activity, although at a slightly higher dose. So this gives us some confidence to pursue further with our compounds. So at the same time, while we are working on the space of Mycobacterium, we also came across with other papers that reported the repurposing potential of nitroimidazole drug in parasite space. So this is something, a new space for us, because bacteria and parasites, they could be very different. But the study that has been done by Professor Allen in, I think in Dundee, they have really basically, they take delamanide, which is a known TB drug, and they test them on Leishmania, which is a type of parasite that called Vaccinia leishmaniasis. And they found that this particular anti-TB drug could be repurposed as a potential treatment for Vaccinia leishmaniasis. So repurposing potential is also a very powerful method where you can use a drug that is originally used for other treatment, but you repurpose them, you reposition them into another application. So this can really help to save a lot of time instead of developing a completely new drug. So this work has inspired us. Perhaps we are thinking maybe we can also test our compound against parasites. In this case, we expand our spectrum. We are not only testing on Leishmania, we're also testing on trypanosomes, another parasite that cause a global issue of the Chagas disease and also the sleeping sickness, which is Trypanosome brucei and also Trypanosome cruzi. So we test them on this parasite as well. At first, we don't get much activity. So at first, when we test them on these 2 parasites, we doesn't see much activity compared to the one we see in Mycobacterium. But then when we further modify the molecule, this time we do see a significant improvement of activity. By just installing an external or an extra pyridine ring in the molecule, we find that the activity significantly improved from previously parasite not active, becoming a micromolar of less than 1 micromolar IC50 value. Although we can see that the Mycobacterium activity seems to be dropped at this time. So meaning that we have basically tuned the activity more towards parasites and away from bacterial. So we further modify again. That's the beauty about some design of molecule. That's the beauty about structure modification. By just swapping the ring around, the pyridine ring, we change it from the first position to the second position. By just swapping it, we find that the activity now is completely changed again. Now the spectrum of activity has become broad spectrum. So we are able to make the compound active against both parasites and also Mycobacterium tuberculosis. So this is the beauty about medicinal chemistry. This is why we like chemistry so much, because when we change a little bit on the structure, we can completely change the activity. We can either increase or decrease the activity. We can also change the spectrum from bacteria to parasites, and we can play around with all those modifications. So something interesting to be noted on here, the previous nitroimidazole drug in the market, the Lemonade, it is active against Leishmania, but when we test our compound against Leishmania in this case, none of them seems to be active. So this tells us that potentially our compound might work differently in parasites compared to the known drug. So this is also something interesting because this can mean that they have the potential to target different mechanisms. So again, we also prove the efficacy of our compound in the in vivo model. So in this case, we took two of our most promising compounds, 73 and 74, from the library. So again, this study, as a chemist, this is not done by me. This is done by my collaborator in UK. So what they did is they treat the mice with a 5-day treatment, in an oral treatment. So they found that in day 24, our compound is basically able to clear the parasites completely in day 24. But in day 36, you can see that the parasites get relapsed. So this is similar to other known drugs as well. So benznidazole is the known drug for this parasite. Benznidazole, as you can see, basically from the diagram there, which is the second column that you can see, in day 18, day 24, they can clear the parasite burden, but in day 36, they also get relapsed. Because we stop the treatment in day 18, and we started to measure the parasite burden. So in day 36, everything gets relapsed back. But nonetheless, it also gives us the potential and some promising outcome that, yes, perhaps we can pursue this further with further modification as well in the future. Okay, so of course, there are still more work to be done here. So we are actually trying to apply some research grant in order to pursue this study because of their interesting results. So we wanted to move forward in the next step is we wanted to test the mechanistic pathway of compound by conjugating the molecules with fluorescence probes, and hopefully we are able to see, to visualize how it basically binds in the target. Either it's in bacteria or either it's in parasites. So, okay, this is done with my first story. So now I'm going to move on with my second story. I still have time, right?
Yeah.
Okay. So the second story, it won't last for too long because this is just a new project that is done by my student currently. So we use quinoline as our pharmacophore to start off with. So why specific quinoline? So quinoline is the one that basically highlighted in pink color. So this ring is very powerful. We call it a privileged scaffold in medicinal chemistry because you can find a lot of quinoline rings in the drugs in the market, in anti-malaria drugs such as chloroquine, in bedaquiline such as the TB drug, in ciprofloxacin, which is something that perhaps you all quite familiar with. So this quinoline ring is very useful in a lot of applications, and the best part is you can see all those R1, R2, R3, R4, R5. Those are the sites that we can potentially modify them. And we believe that each site of modification could lead to complete different activity. So currently, one of my students is working on a quinoline indole molecule. So these hybrid molecules, we take inspiration from indole is because indole itself has originally found to have a very good antibacterial activity. If we modify it differently, it could either act as an efflux pump inhibitor, or it can also inhibit FABH, which is an important enzyme that contribute to the biosynthesis of the fatty acid cell wall. So we then hybridize these two molecules, the quinoline, the pink part, and indole, which is the purple part. We fuse them together through the R3 position from the quinolines. And I'm not going to talk detail into the chemistry part, but I just want to highlight that when we originally designed this molecule, we wanted to make it easy to mate because we wanted to make sure that the reaction is clean enough. We do not require a lot of workup because we want to save time, and we wanted to make a huge library of compound in a very simple way, in a high throughput way. So this project itself can be handled even by honor student, undergraduate student, because we have to design it in a way that it is easily handled by student and using some less hazardous chemical to start up with. So what we found is that the final product, which is we called it the quinoline indohydrazone product, this product is able to yield a single product easily for most of the cases, although it also has the potential to form isomers. So isomers is also another important factor that could contribute to different activity. So there are potential to form the E isomer and the Z isomer for the final product because of the bond could rotate either to form E or to form Z isomer. But in most cases, for our reactions, we found that predominantly E isomer were observed. And this can be observed either by X-ray crystallography, where we crystallize the compound and analyze them by XRD, or we can also observe them by NMR. So from NMR, we see that only single product that is being formed, which is the E isomer. And this is just a very brief SAR data because it's still unpublished, so I will not disclose much about the actual structure, but in general, what we found is that the electronic properties could basically have an effect on the activity. So this project is basically, we aim to target on escape pathogens or some gram-positive and gram-negative bacterial. So we found that the donating group plays an important role to give the activity. When we switch it to withdrawing groups, the activity completely lost. And we also play around with aliphatic chain. Shorter chain is better in this case because when we have longer chain, it leads to poor solubility, and therefore the activity is also not consistent. And surprisingly, when we take out any substituent, we just put NH group there, the activity seems to be not bad, but then it lost its broad spectrum activity. So now the compound becomes only active against gram-positive bacteria, but not against other gram-negative bacteria. So because of the feasibility to synthesize a large library in a simple reaction, so my student also trying to replace this indo with other aromatic substituent. So by just replacing that indo group with other aromatic substituent, we see a similar trend. Again, donating group is important. Withdrawing group lost its activity. And if we have high excessive heterocycles, such as pyro group, for example, activity retained, but when we have pi deficient heterocycles, activity lost. So this again tells us that the electron donating properties could basically play a role where it donates electron perhaps to the quinoline ring and gives a better activity. So we then trying to see if we can identify any potential target for this particular quinoline indo hybrids. So we came across this target, DNA gyrase. So DNA gyrase is a known target for ciprofloxacin and also other fluoroquinolone antibiotics. Because our molecule also having a quinoline ring, so we think that perhaps it could also target a similar potential enzymes. So we then take this DNA gyrase and we try to understand the different subunits. So DNA gyrase has both gyrase A and gyrase B subunits. So gyrase A is the one in the blue color, as you can see. It represents an important role in binding, cleaving, and rejoining DNA during replication, whereas the gyrase B subunit is the one colored in pink. It's usually possess the ATPase activity. So we see that the ciprofloxacin itself, they do have an important role with a different modification. So when you look at the position 7 in the ring there, the position 7 basically is the one that is going to be involved in the binding more towards gyrase B, and whereas the carboxylic acid group, which is closer to the right-hand side ring, it's interacting more with the gyrase A. So we then perform an in silico analysis. So this in silico analysis is basically help us to predict the binding affinity of our molecule. So we first trying to dock our molecule into the ATP catalytic site. So we found that this Indo-quinoline hybrid, when they dock into this ATP catalytic site, they seems to have quite comparable energy, which towards the binding affinity towards the cavity, similar to the control dock. And we also see that the quinoline ring itself perhaps is quite important because this is the site where it involves more affinity or more interaction with the amino acid residues that is known to important for ATPase activity and also DNA supercoiling activity. So we also found that the Indo ring parts, which is the one in blue part, it's mostly more involved in the coupling of ADP hydrolysis. So again, we have performed another, I think that's some issue that I can't switch my slide. Okay. Yeah, okay, that's it. This is perhaps close to the end. I think my slides also need a rest. This is, I think, among the last, maybe the last two slides. So as mentioned earlier, the previous slide, I've shown you that the binding towards the ATP catalytic site. And then we also trying to dock our molecule into another DNA cleavage catalytic site. And in this time, in this DNA cleavage catalytic site, we see that the binding energy is not as good as the ATP catalytic site. So this could means that our molecule, they probably bind better to the ATP catalytic site instead of the DNA catalytic cleavage site. But again, we still see some binding affinity, but the G-score, which is the binding energy, is not as good as the control, as you can see here. So we are still trying to use this docking results to help us to basically design new molecule, because from the docking result, we will see that by changing some structure modification, we can either increase the affinity or decrease the affinity. So this can help us to further design the molecule in the future. And we are also planning the next step forward is to test the compounds in the enzymatic assay. So we are trying to purchase the DNA gyrase or the DNA gyrase kit, but it's quite expensive. So we are still trying to figure out on the financial part. So this is the end of my talk. I would like to just take this opportunity to thank everyone involved in both the projects. Of course, the Queensland team is the one that I still keep a good relationship with. That's my previous supervisors, Mark, and also other team members who helped me in the nitroimidazole project, and also the hybrid compound on the quinoline side is mostly done by my student in Monash. I did not do any of this work. This work is all done by my students, Kwang Kim, Shuwen, and also Jiwoo. They helped me a lot in the project. And thank you so much. And if you have further questions, please feel free. Thank you.
Thank you. Oh. Stop sharing. Okay, maybe we'll go like this. There you go. There. Okay, thank you, Dr. Chee, for that wonderful presentation. It actually takes me back personally to PhD days as well, looking at this presentation. Yes, I think we have maybe about 15 or so minutes for question and answer session. If anyone would like to ask question, I will probably open the floor for the offline participants first, if anyone would like us to ask any questions. Not yet. Oh, you do? Okay. Hello. Nice to see you. You're back. So I will just introduce you. Okay. So we have a question from one of our offline participants. Maybe you want to come on this. Oh, over there. Yeah. No. Yes. So. And then where are you from? What your name and your institution, and then your question, please.
Thank you. My name is Andri. I'm from Soekarno. This is Center for Genetic Engineering, BRIN. So actually, thanks for the slides. I'm not a chemist, but I did my bachelor. I did chemistry in my bachelor. So actually, my master and PhD is I always working with chemists because I'm working in protein drug conjugate. So I didn't get
Okay.
So you mentioned about PPD, about linker. I tried with antibody with PPD. It's really very toxic, right? But when I think it also depends on the linker.
Yes.
When I try it in vitro assay, it's not toxic as like I expected. So the problem, I mean, I'm not a chemist. I'm working in antibody drug conjugate, but my focus is on specific conjugation. So like which site for the drug conjugate. I mean for the English efficacy, right? But I think the problem is always the linker. So you mentioned about the linker, like the cleavable linker. I tried like Ph.D. That's a cleavable linker. But I found some journals they prefer to use the non-cleavable as well. I'm not working for the antimicrobials, but in general for the anti-cancer agent. So it's very challenging. For me, I mean, I was working with the chemists, but like my colleague is organic chemistry. She always tried to design the I think the linker is very important in our case. So can you explain your experience?
Okay, sure. Thank you, Andri. It's nice to see you again. We have met each other in UQ many years ago. For my case, it depends on what type of project I'm working on, because I also have a project working on protein and drug conjugate. And also I have another project at small molecule and small molecule. So maybe I talk about small molecule and small molecule drug conjugate. So we do study a lot of different linker, as you mentioned. Linker can be a major issue. First thing is that the length could be a problem. If the length is too short, perhaps sometimes it will mask each other because they have a close proximity. Sometimes if it's too long or if it's too hydrophobic, it could also cause solidity issue, and there are many, many more that we need to further investigate. So therefore, we sometimes come across what we call structure-activity relationship, where we try to have design like quite a number of molecules and see which one fits the best, because I believe different target is all different. So we really have to do more in-depth study. And regarding your mentioning about the cleavable and the non-cleavable linker, I think for hybrid drug, it depends. Because hybrid drug, even if it's a non-cleavable linker, it could also does the work because it can make it a new molecule itself, where it will exert the activity as a whole thing. And if it's a cleavable linker, it could act as a prodrug, means that when they go to the specific target, they get cleaved off and individually react to one another. I do have more problem with the protein drug conjugate. So I think similar with your antibody drug conjugate, where in sometimes because the drug is so small and the protein is so big, and you don't know exactly where it's the potential site that it goes on. So this is also something challenging. And I'm still trying to figure out this problem similar to you. So I can't really help you much in that sense.
The best side. In the cancer side, I guess I'm from this country, you know well, right? That's what I meant. So that's very true. And also, you mentioned
Yes. Oh, that is actually not the work done by myself. It's a reported literature. Yes. They do conjugate PBD with ciprofloxacin, and they see that it can reduce the cytotoxicity of the PBD.
maybe one more. Actually, CP, in my research, our research group, we on CP as well.
Yeah, okay.
I mean, just like we are in engineering, so we work on manufacturing and power collecting. Children are really working. CRM also the technology. So when working on the, I know Professor Nicholas.
Yeah, yes. Oh, okay, okay.
Yeah.
It's good to know.
Yeah. Now we got
Okay,
yeah.
okay.
Yeah, I think this has been your like working on small molecules.
Yes.
You know, we have the self-link.
Okay.
I mean, we're working on that, everything about the viral devices.
Yeah.
Yeah. So
I'm definitely interested to collaborate further because sometimes you have to send compounds to Australia could be a problem as well. Yeah, if we can do it, maybe that's yeah.
I think I heard there's a technician, they coming from the big plants.
Yeah.
I think they're working on TV before, so now they're working in the software.
Okay, yeah, I need to double check with that because I think as far as I know, they're not doing any sort of TV work yet, but I mean, it is possible to have a look and see.
They have experience before the plant.
Oh.
So, yeah.
Yeah. Thank you, Andre. Okay, thank you for that, Andre. And I think we have a number of people who've put their hands up. Okay, I will let Bapak Ferdiansyah Anugrah, if you can please ask your question, give raise your hand, please ask the question, your name and your institution, please. Thank you.
Okay, thank you, Miss. Hello, my name is Ferdi. I'm from Sultan Ageng Tirtayasa University, Cilegon, Banten, Indonesia. Now I'm studying at Chemical Engineering Department, which my focus thesis about antimicrobial from metabolite secondary endophyte microbia for bioinhibitor and biocoating. I am really curious about for your research, Miss. I want to verify, have you made the differential equation model for your research, such like correlation between activity and solubility? And if you make the differential equation model, what book or maybe article or even additional researcher you use for when you make differential equation model? Thank you.
If I understand correctly, sorry, because I don't really hear very clearly about questions, but if I get it correctly, I think you're asking about the correlation curve of the activity versus solubility, and how do I get this correlation curve? Is that correct?
Yes.
Yeah, okay, so basically the correlation curve, what I did is that for the y-axis, which is the activity that represents the MIC against the mycobacterium, what we did is we convert the MIC value to a PMIC value, and then from the x-axis, which is the solubility, we basically experimentally perform the solubility assay by ourselves, where we use liquid chromatography to basically find out the solubility in aqueous, and then we get the solubility and we convert that to the log S value, and then we use that to plot the curve and get the correlation in order to see how the activity and solubility, what are their relationship to one another. We don't really have a specific model to fit in this case, because we just wanted to see the whole picture where the insoluble compound or the soluble compound, whether they will contribute to differences in terms of the activity. Yeah, I hope that answers your questions.
Would that be sufficient, Ms. Ferdiansyah?
Okay, so when you you get MIC value, you convert to PLOG, ya, Miss.
Yes, yes,
Not. Oh, okay, okay,
yes.
okay. Thank you, Miss.
Thank you.
Thank you for your question, Ms. Nidyanshu. I think let me see if we have any more questions. Okay, so it looks like we have a question from someone offline as well. Please state your name, your institution, please. Thank you.
Always have I did school leadership and I really want to know like how do you decide the looker?
Yeah.
Like you start with
Yeah.
It's like how do you decide the looker between the two parts?
Yeah.
Then based on what I get it.
Yeah.
By which looker.
Yeah.
And the second one is like always consider solubility and this other skills related to plants.
Yes, yes.
So and we are tested several students, I guess.
Yes.
And when that mixture drugs to the mice, may I know that what do you use like intravenous or is oral?
Can you tell me more detail about it?
Apologies. There were some technical issues, so we couldn't actually hear you in the Zoom. You're asking a question. So I will probably rephrase your first question. So we have Fitri asking about what was the reason, or what sort of criteria did you use to select linkers to connect between the two molecules that you're choosing? So that was the first question. And can you please repeat your second question?
Okay, the second question is, do you always consider the solubility and liquidity for your drugs? And I assume that is for drug delivery. And you already tested some of your drugs to mice. So I wonder that how you administer the drug, is it using intravenous or using oral? So can you more explain about that? Thank you.
Thank you, Fitri. I'll answer the first question first regarding the choice of linker. I think the choice of linker depends on whether you want the drugs to be administered at the end as a prodrug or as the whole new molecule, as a whole thing. So if you want the molecule to stay intact together at the end, you will design a linker that is stable so that when even administered to the site of action, it is still remains stable as a whole molecule. But if you wanted to make it a prodrug where it will split into two different molecules, then you will design a linker that is going to be, for example, hydrolyzed by enzymes, or it's going to be cleaved off under specific pH conditions, so it gets cleaved off at the end. But of course, how do we find out at first? First, as I said, we will see which one we are targeting on. And the second is that we will see, of course, another issue is the accessibility of the site of modification. So sometimes a specific linker could be more difficult to make because we might need to modify the functional group of the particular drug in order for them to link together. We can't change too much on the drug as well, because if you change too much, it can also lose activity. So we try to see whatever functional group that is first accessible to the drug, so we can link them together. Or if there is no any other functional group, then we will have to start to think about how to modify them. And then once we get this done, then we have to design how long is the linker that we want to make. So currently, my student, the second project that I show, we haven't explored much on the linker effect yet. We're just trying to see these two pharmacophores first when we combine and link them together. Are they good? And now the second approach actually we're trying to see is to understand different linker effect in terms of their lipophilic linker or polar linker, the length of the linker as well, and also whether we want to make it a cleavable or non-cleavable at the end. And for your second question, I think you're asking, my memory is bad, sorry, you're asking about the oral or IV administration, right? Yes, yes.
These two criteria is really related to membrane permeability, right? I assume that this is about the drug delivery itself. So it's like, I wonder that when you are tested in the mice or in other phase, clinical phase, do you administer this like intravenous or via oral? Because it's like, if it's already on the market, the drugs will be in various type, right? Like tablets, capsules, and so on and so forth.
Yeah.
So I really want to know how you administer.
Okay. So for ultimately, that could be given via orally, because it's more convenient way to administer to the patient. It saves out a lot of cost, and also easy for the patient to uptake the pill itself. So originally, our idea is to get oral treatment, but you can see from my result, solubility can be a main issue. But then when we test them on the animal work, on the mouse model, we still try oral administration, but we have to do some formulation. We still have to undergo some formulation, for example, use PG to basically help us to better solubilize. So we use that and administer in oral for both TB and also for the parasites work. Yeah.
Or like personally, or
yeah.
017.
Yes, of course. Yes, formulation is not my strong forte, but I do see that sometimes we still need to improve the formulation of a particular molecule in order to exert its administration way and also to have a better pharmacokinetic profile. But I think for my stand is that we will always try to design a molecule first to make it at least have the good property before we think about the formulation. So the drug molecule itself has to possess a good drug-like properties first before we think about formulation at the second part. Yeah. Thank you for your question.
Okay. Thank you, Fitri, for that question. And probably I would like to maybe close for this particular talk for now, in the interest of time. And I would like to thank Dr. Chee for a lovely presentation, and yes, so I think now it's time to move to our second presenter, Dr. Ken. Yeah, this one as well. Oh, yes, yeah. It's actually limited. Just bear with me for 1 second. Yes, this is working. Oh, yeah. Good? Yeah, okay, so myself. Maybe I'll pick this one up. This turn. Okay. All right. See. Okay. Thank you everyone for continuing to be with us for the second part of this webinar. So I would like to invite Dr. Ken Yonyong, or Dr. Ken. Hi Ken, how are you? Hopefully you're good. And before we proceed to his talk, I would like to briefly introduce yourself. Introduce to you, Dr. Ken. I'm a little bit, yeah, I need some coffee. Anyway, so Dr. Ken is currently a senior lecturer and the associate head of School for Industry Engagement and Enterprising, the School of Science, Monash University, Malaysia. And his research focused on small molecules and short peptides in drug discovery, cancer, and neurological disorders, as well as infectious diseases. So we're actually branching off in a way from most anti-parasitic drug design from Dr. Chee's talk to potentially having a broader peptides-based research sort of sharing session with Dr. Ken. currently has co-authored over 80 research publications in the field of medicinal chemistry and drug discovery. And he's also has been awarded a number of awards and grants supported by the Horizon Europe as well as the Ministry of Higher Education. And he has quite extensive patents applications and multiple academic awards. And he completed his PhD in medicinal chemistry quite some time ago from the University of Science Malaysia. And he has since had quite an extensive research experience as well as a research scientist in rhinogenics and also the School of Science, Monash University, Malaysia. And I'm sure that his talk today will also, similar to Dr. Chee's talk, will also give us some ideas and potentially have, give us ideas for potential potential collaborations in the future. And with quite an extensive awards and honors that he has so far, with different teaching excellence awards, as well as supervision awards and top 2 scientists recently from Stanford and Elsevier. So big round of applause. And quite a lot of publications in highly reputable journals. So I'm sure that Dr. Ken here is an excellent resource person to have with us talking about peptides-based research. And it will be an interesting talk, I'm sure, for all of us. So with that, I would like to invite Dr. Ken, and the time is yours. 45 minutes, and then we'll follow with questions.
I'll just probably share my slides first. Oops.
This one? I think it's the text. Is this the one in this? You can open this one. And then I'll go to the text.
Can everyone see my slide offline? Oh, online, sorry.
This is
Loaded.
oh wait. Where's this? Yeah, I think it's already. This one. This one. It should be this one. It's still not going. Just try to open this one. Yeah, and then maybe maximize it and share the screen.
Okay.
And
Yeah.
Sure. Okay.
All right. Thank you very much, everyone. And thank you, Dr. Anja, for the very kind introduction. Today, I will be sharing some of my overview of my research in terms of how our labs basically exploit small molecules in terms of drug discovery. But before I go into the actual technical aspects of it, due to my current role as the Associate Head of Engagement, please allow me to share a little bit of information about Monash University, Malaysia, and specifically the School of Science. So Monash is basically a global university, and we have 9 campuses around the world. Malaysia is the 3rd largest campus after Clayton and Caulfield. And the newest addition to Monash is Monash University Indonesia, which is currently situated at Ph.D in Jakarta. Right? But of course, Monash University Indonesia basically is a campus that focuses on the postgraduate studies in business as well as information technology and architecture. So this is Monash University, Malaysia. I think some of you have basically been to Monash University, Malaysia. So aerial overview. So around the perimeter highlighted in blue over here, this is Monash, but the landscape has probably changed quite a little bit. We do not have the green lung anymore, unfortunately. It has been replaced by the student hostel, brand new student hostel, which is very nice. So basically, I'm located, or School of Science is here, and I'm located at the level 8 top level over there with a nice lake view. But of course, the lake doesn't really belong to Monash. And Monash University, Malaysia has approximately more than 9,000 students currently, and the School of Science is a medium-sized school where we have approximately 10% of the total student population of Monash University, Malaysia, with 900 over students, with 34 academic staff, 3 research fellows, and 24 professional staff, and over 4,000 alumni at the current moment. So in the School of Science, we have 5 research teams divided into chemistry, drug discovery and development, tropical biodiversity and conservation, genomics and big data analytics, functional and future food. And some of us, for example, Chee and myself, we work cross-disciplinary between chemistry and drug discovery. So an overview of my group's research. So we consider ourselves as a drug discovery and medicinal chemistry group where we use chemistry as a tool to solve biological problems. And at the heart of our research is technically the synthesis, right? So 2 very broad areas that our lab works on. First of all is the synthesis of bioactive small molecules in drug discovery, and that also includes short peptides, as well as synthesis of fluorescent molecules for diagnostic application. So at times we do come across molecules which may have both properties, for example, the therapeutic properties as well as the diagnostic properties, and we can then use them as a theranostics, right? So if we have the same molecules, then that's basically a very interesting molecules where we can also do therapeutic as well as imaging at the same time, for example. So when we do drug design, maybe just a little bit of background over here, which I think is relatively relevant. So when we do drug design for small molecules, for the past 30 years or so, most people actually started off with following Lipinski's rule of 5. So this is a broad guideline that is developed by Christopher Lipinski when he was working at Pfizer. He and his team basically analyzes more than 2,000 mammalian drugs, and what they found out is a set of rules or guidelines that can predict whether small molecules will be drug-like and have good oral bioavailability if they actually have these 4 criteria, right? So molecular weight less than 500, no more than 5 hydrogen bonding donor groups, no more than 10 hydrogen bond accepted groups, and a log P of less than 5. So that has been the cornerstone of the design of small molecules in order to possess drug-likeness and oral bioavailability over the past 30 years or so. But since that publication, quite a number of things have changed over the years. One major highlight is in terms of the molecular weight that the current drug actually has. So when Lipinski and his team basically worked on the analysis, as you can see prior to 1998, in the blue bar chart, you can see that that kind of prediction actually holds true when majority of the orally available drugs are less than 500 Dalton. But Since then, since the turn of the century, we can see that majority of the small molecule approved drug that is orally bioavailable, they have grown in size. So now, majority of them actually concentrates at about 400, 500, and even up to 600 Dalton. All right. So we to go with the time, we probably need to tweak a little bit how we actually design a good small molecules. And there are also some other criteria that basically can be incorporated nowadays when we have more information, how we can basically design these molecules. So at present, we determine or we term small molecules as something that is less than 750 Dalton or 750 grams per mole. So it has moved from 500 to about 750 right now. We also think that a drug would have desirable properties if it has a high fraction of sp3 carbon. What it means is that it will have a more 3D-like structure to basically bind in the active site of a protein. Low aromaticity. So we have to be a bit careful when we design our molecules as well. So low aromaticity over here basically means that if we think that a molecule is too flat or too planar, we can replace, for example, the benzene ring with a cyclohexane. Reduce rotatable bonds. So what we did was also to change some of the linear structure of the compounds and recyclize them, and we use things which are a little bit more cyclic, like a 5-membered or 6-membered cyclic ring. And the best if it falls between a log P of 2 to 3 if we are targeting mammalian diseases. All right, meaning that it has to have a balance of lipophilicity as well as aqueous solubility. There are also some others that has been proposed previously by Lipinski, such as how many hydrogen bond donor and how many hydrogen bond acceptor, which has not changed much over the years. So this is also some of the criteria that my lab uses when we design some of our synthetic compounds, especially for compounds targeting the age-related or the mammalian diseases. So in terms of target diseases, these are the few target diseases of interest in my lab currently. So we do a little bit on antimicrobial, on anti-cancer, as well as on neurodegenerative diseases. But before I move to antimicrobial, please allow me to start with anti-cancer, because that's where my lab's research actually starts from. And I also understand that there might be some interest in BRIN on anti-cancer research as well. And after I've completed the anti-cancer slides, then I'll move to antimicrobial. Our interest in small molecule cancer drug discovery actually stems from our interest to look at sirtuin enzymes. So sirtuin enzymes are enzymes that have the acetylase or monoacetyltransferase activity. And currently there is 7 human mammalian sirtuins that is decoded SIRT1 to SIRT7. So it's a relatively new target because it has only been discovered in the 1990s. It's part of a HDAC group of family of proteins. But the difference is that for sirtuins, it doesn't use zinc as a cofactor, but it uses the NAD+. So that would also serve as a rationale in terms of how we design our drugs targeting sirtuin later on, which I'll show you. So what is so interesting about this group of new, so-called new proteins at the turn of the century is that it is implicated in many age-related diseases, such as neurodegenerative diseases, cancer, diabetes, and cardiovascular diseases, right? So it kind of serves as a master enzyme that control some downstream targets of all these diseases. So if you could basically target sirtuin, technically you can control some of the pathways that is related to all these diseases. But what actually created the spark or the main interest in sirtuin research is the discovery of resveratrol as a SIRT1 activator. All right. And SIRT1 activator has been touted as a potential anti-aging agent. So that is really big news then. In 2003, there is a lot of publication, especially in Nature and Science, talking about how sirtuins can potentially become an important direction of research in anti-aging over here. And at layman terms, of course, this is also very exciting to people, because if you tell people that you can just pop in a pill and then you can extend your lifespan for several weeks or several months or several years, that seems pretty exciting to people. So people actually get on the hype of sirtuin and resveratrol being able to extend lifespan or anti-aging. And that has also been captured by even the Fortune magazine, where they put up a front page over here where it says "Drink wine and live longer." So we have also quite a number of prominent researchers working on sirtuins and anti-aging, especially to note Professor David Sinclair from Harvard University, who is actually a co-founder of a company called Sirtris that basically works on synthetic sirtuin activating compounds, or what we call STACs. And in 2008, together with his co-founder, they have sold off Sirtris, the company that he founded, for $720 million to GSK. So this shows that if you found something really interesting and if you really have good compounds, then that can basically make you very rich, right? But we know from evolution, human evolution, that it is always quite difficult to activate a protein compared to inhibiting a protein for biological processes. And with that, the hype of sirtuin activator as an anti-aging actually died down, because there's some issues to that. But what is certain is that if we're able to inhibit sirtuin, especially SIRT1 to 3, it potentially can be a very good anti-cancer agent. And that's how we come up with our first discovery of benzimidazole being used as a sirtuin inhibitor. So we are the first group that basically report on that. So it started off with in silico prediction of a library of compounds. And then we think that from the first top 100 that we obtain, the structures that we obtain, benzimidazole basically ranks quite high. But again, this just serves as a prediction. So after we have done the prediction, we do also carry out some analysis for us to think whether it's worthwhile for us to carry on with the project. So why benzimidazoles? When we do some analysis, as mentioned earlier, NAD+ is a cofactor of sirtuins. And over here on the left, we have also EX527, which has been reported earlier as a very potent sirtuin inhibitor. In terms, when we compare these two molecules, we can see that there's some similarities, especially those highlighted in green and in purple over here. So EX527, which is synthetic, it's kind of like a hybrid or a mixture of the two terminals from the NAD+ structure. And benzimidazole over here is relatively similar again to the structure highlighted in green for EX527. as well as NAD+. So we think that with these similarities, there is potentially some good overlapping in terms of the chemical structure that we can use as a base to start off to test whether it's a good inhibitor for sirtuin. This is agreeable with the in silico data that we obtained earlier. So we think that, okay, this might be a good go ahead to start off. So we did about 200 different derivatives of benzimidazoles based on the in silico prediction. And we come up with BZD9L1, which is the best and most potent sirtuin inhibitor that we have at that time. So it is a semi-selective SIRT2 inhibitor. As you can see, for SIRT2 is about 9, IC50 is about 9 micromolar, and SIRT1 is approximately 43 micromolar. So it's a semi-selective SIRT2 inhibitor. At that moment, these values, of course, at the current moment, it's not something spectacular because we have already much better certain inhibitors that has been reported. But at that particular time, approximately 10 years ago, that was as good as what you can find on the market. So we are quite proud of this achievement. So in terms of the synthesis, it's actually a 5-step synthesis. I'm not going to go through that today. But what I want to highlight over here is that you can see in terms of the final scaffold that we have, there is many substitution that we can put into the structure, especially at R1 and R2 over here. So it's very modular. So that's how we basically come up with the first initial 200 structures by modifying these two groups over here. And having good sirtuin inhibitory activities does not necessarily mean that it's going to have a good anti-cancer activity. Of course, it's predicted to have, but then we wanted to confirm using cell line assays as well. So we have tested on colorectal cell line, HCT116 and HT29 over here. And we can see that the IC50 is approximately about 16 to 20 micromolar, which is pretty decent. But what is important or interesting over here is that the compound BZD9L1 actually has very good selectivity against cancerous cells. This is important because when we further develop the compounds, we do not want to have a compound that is also toxic to the non-cancerous cells. I mean, that would be detrimental, right? So we want to ensure that it only kills cancerous cells, but not the non-cancerous ones. So far, it seems that we have pretty good data over here. And another interesting aspect of BZD9L1 is that it's autofluorescent. So it has approximately 23,000 in terms of the epsilon value and quantum yield of 33.7, meaning that it's quite highly fluorescent. As you can see, it fluoresces in blue over here. And we actually uses this compound as a theranostics to image how these compounds penetrate into the cell and where it's localized in the cancerous cells. So this is actually quite interesting as well. We did a little bit further studies on the mode of action, and we find that BZD9L1, which is the hit compound over here, actually leads to early and late apoptosis. So this is what we think actually is happening and how this BZD9L1 actually kills off the cancerous cells. But we are not at the end of the story because we think that there could be further improvement to the compound that we have at that point in time. So we go back to the docking and we find that the BZD9L1 actually overlaps with the NAD+, which is the cofactor for sirtuin. We think that it actually overlaps with NAD+, and kicks out the NAD+ from the actual active site when it works in the protein. So this serves as a prediction that it might actually be a competitive inhibitor. So we do some assays to confirm that this is so. And we find that with increasing concentration of NAD+, the SIRT2 activity actually decreases. So this kind of confirms our earlier prediction of it being a competitive inhibitor. So with that in mind, we know what are the amino acids that is lining the interaction between the molecules and the protein. And with that information, we can actually design better compounds. The second generation of compounds that we have is by further modifying the structures highlighted in red over here. And with the modification, we come up with another compound, which is PZD9Q1, that have anti-proliferative activity or anti-cancer activity between 1 to 10 micromolar. So we managed to kind of reduce the IC50 over here. In terms of SIRT2 inhibitory activity, also much better compared to the earlier version of PZD9L1. But then we can see that it loses the selectivity or semi-selectivity of PZD9L1 against SIRT2. This could be a good or a bad thing depending on the cancerous cell lines that we are interested in. And further modification, this time on the left-hand side of the structure, highlighted in blue over here, of the ester group, because we know that esters might not be so stable, metabolically stable in vivo. So we change the ester into some other groups which are more stable. And we are delighted to see that the anti-proliferative activity also reduces. It means that the IC50 also reduces. Now it's at a sub-micromolar level, where we are quite happy at the current status because we think that it's good enough to go into animal studies right now. And some of these new derivatives are also highly fluorescent. Again, we can also do some imaging with these compounds. Some transcriptomic studies, we did some string analysis as well to determine some pathways over here which are related to the activities of the compound. And then we try to confirm the pathway by the qPCR as well for the gene expression, as well as the Western blot for the protein expression. So this work is still currently ongoing. Another thing that which I want to highlight over here is the new project that we have with Cancer Research Malaysia, as well as University of Swinburne. It's on the ADAR1 inhibitors. So we moved slightly away from SIRT2 to ADAR1 inhibitors because from a previous publication, previous research from our colleagues from Cancer Research Malaysia, they have found out that the ADAR1 is basically a key driver gene for the survival of oral cancers. So I think that's where moving forward, that's where the funding is going to be in terms of the funding, where they look a little bit more closely on the specific demographics, for example, like in Malaysia or in Indonesia or in a specific Asian countries. So we think that that's where the funding will actually come from, rather than a broad, general, worldwide kind of research on cancer, because that for cancer is a very complex disease and there's no one size fits all. So we are moving into a little bit more specific population when we're working with cancer over here. I think that's basically a little bit of glimpse of our interest in anti-cancer small molecules. And I would now like to move into antimicrobial, which probably would interest some of you over here as well. So when we talk about antibiotics in general, we have a different trend or a different strategy compared to the ones that we used to do for anti-cancer. Because for antibiotics, we know that usually does not follow much Lipinski rule of 5, where in a Nature Review Drug Discovery article over here, we can see that majority of the antibiotics are on average, they are more hydrophilic and slightly larger compared to those targeting the mammalian diseases such as central nervous system. So in terms of the Lipinski rule of 5, we don't really need to follow as closely for antibiotics. So that's what happens with our group's research in antibiotics as well. And we also do phenotypic screening rather than target-based screening. So that's again another difference for antibiotics in terms of approach from our group. So if we do not use in silico prediction or design, so how do we actually come up with the scaffold of interest over here? Most of the time for antibiotics, we rely on natural sources because we think that natural sources are a good pool for antibiotics. From a report in 2020, it was reported that there's from the approximately 180 anti-infective drugs that's commonly used, 163 of them are derived from natural compounds, which is a good proportion, and 94 of them are actually antibacterial. So it's kind of like a good way to start to get the scaffold of interest. But it's not only plants over here that can basically be of interest. It can be fungi, it can be algae, it can be bacteria. So all of these are good sources of natural sources to start off to get the scaffold of interest. So in our lab, we have 3 different strategies for antibacterial agents, especially those focusing on small molecules. We do have single scaffold molecules, hybrid compounds, which Dr. Chee has mentioned a little bit just now, as well as short peptides over here as well. So for single scaffold molecules, our approach is basically on semi-synthesis of bioactive compounds. So usually the compounds of interest will actually stem from some exotic plants that our collaborators have actually collected, extracted, and isolated. So they know that which of these compounds is actually exerting the antibacterial or antimicrobial activity. So our group's position in this kind of project is basically on the semi-synthesis, where we uses a parallel synthesis approach to basically modify the molecules, the initial molecules, which is active. So our idea or our aim is to improve the pharmacological properties of these isolated compounds in terms of potency, in terms of solubility, in terms of stability. So that's what our group is actually doing. So we also have short peptides over here. In terms of short peptides, we are focusing on peptides, which is about 5 to 15 amino acid in length. The reason why we are not focusing on very long peptides is the fact that if a short peptide can basically do the job as good as a long peptide, a short peptide would be more feasible due to the synthesis of the peptides as well as the cost of the peptides. When the peptides gets longer, usually it gets entangled when we do the synthesis. and there is always an issue of trying to hit the terminal to increase the length of the peptide. And stability of the peptides also may be compromised when we move above, let's say, 30 or 40 amino acids. So if we can do the job with short peptides, that's actually more desirable. And we try to design our peptides based on cationic peptides. What it means is that the side chains of the peptides, usually we try to have those amino acids such as glycine that have amino groups on the side chain. At physiological pH, they will actually be protonated and they'll be positively charged. And that will actually be very good if we are trying to target the gram-negative bacteria, because gram-negative bacteria on the surface of the membranes is usually negatively charged at physiological pH due to the abundance of carboxylic acid groups. So that's one of our strategy. Another strategy is trying to cyclize those short peptides, because we think that with cyclization, where you have a more 3D-like structure, it will actually penetrate the membrane a little bit better as well. So some of the idea of where we get the short peptides over here is from a human virus, for example, the TET1 HIV virus. We get some sequence from that protein, and we selectively try to see which are the region of interest that actually exerts some activity. We have also looked at short peptides that is derived from reptiles such as frogs, frog skin. So that is also something of interest. The approach that we use in terms of the synthesis of those peptides is solid phase peptide synthesis, SPPS in short, which is a very generic synthesis for peptides. We do not have a peptide synthesizer, so therefore we have to go back to the conventional way of adding in one amino acid one by one. So first of all, we do the protonation, the protection of the FMOC group of the amino acids, which has been linked to a solid phase support. And then at another hand, we activate the carboxylic acid group of the amino acid, which we want to add into the peptide chain. And then with coupling, what we did was we uses a coupling reagents such as HBTU or carbodiimide. And after the coupling has been completed, the process is repeated again until you reach the peptides that you want. The final step is on the protection and cleavage, where we uses the trifluoroacetic acid to cleave off the peptide from the solid chain support. So this is one of the methods that we uses to build our peptide molecules. As I've mentioned earlier, if we go towards longer peptides, this process actually gets more difficult because we need to deprotect the terminal. So when you have long peptides, this group might actually be hidden inside the cavity of the long peptide if they actually gets entangled. So we have to be very careful over here. And last but not least is on the hybrid compounds or small molecules as antibacterial agents. This is the one that is the most successful strategy so far for our small molecules as antibacterial agent. So what we did in our lab was to use nature-inspired molecules which has been semi-synthetically modified, and then we hybridize it with a antibiotic, a common antibiotic. The antibiotic chosen usually are those antibiotics that are past their prime. So past their prime basically means that they are maybe no longer in use or less in use, because we try to salvage whether these antibiotics can be, retooled to become an antibiotic that might actually target a different bacteria, for example. So when we hybridize semi-synthetic nature-inspired molecules with antibiotics, such as, for example, ciprofloxacin, we actually get very good activity with an MIC in picogram per mil kind of level. We have not come across many small molecule antibiotics that is at this level of MIC. So we are quite lucky and quite surprised to see this in the first place. And then I asked my students to repeat it multiple times, and different students repeating it. And then we sent it to Singapore for our collaborators to test on it as well. And luckily, the results come back as in reproducible. But this MIC value over here that is very potent, it is selective only to some gram-positive bacteria. It's not a broad spectrum compound. It is very selective towards certain gram-positive bacteria, such as S. aureus and Bacillus cereus and some other gram-positive bacteria. In terms of gram-negative bacteria, it is not exactly that great. It is still active in a single-digit microgram per mil level, but not as active as the against gram-positive bacteria. It is also active against drug-resistant gram-positive bacteria, such as MRSA, which is also of interest in our group, right? In terms of selectivity for this new group of compounds, of hybrid compounds, that because of its low MIC, the selectivity against bacteria is also very, very high. It's more than 1 million times more selective against bacteria compared to cells. So we are quite confident that this type of molecules will only selectively target bacteria and not do any harm towards humans. Because this is currently under a patent application, so I could not divulge that much on the structure of the hybrid compounds that we have, but we are open for collaboration. If anyone is of interest to move this to the next level, for example, in animal models, we're happy to discuss this further. So I think with that actually sums up my presentation today in terms of giving you an overview of how our group actually exploits small molecules as antibacterial as well as anti-cancer agent. Last but not least, I would like to thank my group members and collaborators from these universities listed over here, because I don't do this alone. Basically, majority is my group's work. I would like to thank all of them and the collaborators, as well as the funders from the Ministry of Science and Technology and Innovation in Malaysia, as well as the Ministry of Higher Education, Royal Society of Chemistry in UK, as well as the Horizon Europe. With that, I thank you for attention. Terima kasih. If you have any questions, I'll be happy to answer them. Thank you very much.
Thank you. Thank you, Dr. Ken, and that was a great talk. And again, I think some parts of it reminded me of some of my master's work actually so far, because I actually did quite a bit of solid phase peptide synthesis back in my master's. But yeah, I guess in your talk that you're targeting like you're working on anti-cancer and then moving on to antibacterial drug discovery is something that would be of interest to quite a few people in this room as well as online. With that, I would like to open question session. And let me just stop here for now. Okay. So first of all, since I don't have currently any question from the online participants. And is there anyone from offline? Okay. So we have a question. You might want to unmute. Please state your name and your institution, please.
Thank you. So my name is Andi. I'm from the Center for Genetic Engineering with the cancer drug discovery for the therapeutic agent.
Yes.
So when you synthesize the drug, did you do on purpose to make a progression? Because I mean, the most important thing is the drug itself, right?
Yeah.
And then the second person has been tried in people, say.
Because if there's a therapeutic agent, we can track, right? I mean, if for example, if we make a xenograft mouse models, we can see where is the drug itself. So if there is not agent, maybe we can say, oh, it's very toxic. We can see if the mice has a very high weight loss, something like that. Yeah. So because in my experience, when I want to make a therapeutic, I have to make it two agents. So one have to conjugate with the fluorescence agent, and then another one with the drugs. So we have to make like more complicated, right? So two agents. So I think it's very interesting if you can find the drug with I mean, it's able to do the diagnostic and diagnostic agents. Yes.
Thank you very much for your questions. Very good question. The first question is whether we on purpose, whether we synthesize that. I would say that it's both. Sometimes we know what kind of molecules would basically give rise to fluorescence, and we kind of try to incorporate that into our structure. But there could be also times where it's accidental, meaning that when we design the compounds and we think that it has good therapeutic activities, we also find out that it's basically autofluorescent. So it can be both ways, right? It's not always on purpose, and sometimes it's accidental. In your second question, as in whether we have tried it in vivo, we have not, because our previous compounds, we don't think that they are attractive enough to go in vivo. But now, since we have got quite decent potent compounds, we think that we are ready to probably go in vivo.
So for the small molecule, you do this xenograft as well to make a xenograft mice models for
we don't do that in our lab, so we can basically collaborate if there's anyone of interest.
Yeah. So for small molecule, you always, I mean, when you inject the drugs, do you use the delivery systems or mix with the other delivery system, or just like naked drugs? I mean, just the small molecule. Get the small molecule.
Yes.
So without any delivery system.
No. Yeah. Because we think that it depends, because we have not gone into animal model. If we think that it may need a different delivery system, such as the liposomes or things like that, then we could probably look at that as well. But at the current moment, because we have only tested it on cell lines, we are just using a plain compound. Yeah.
Interesting. It's like, in general, like, if I use the liposome, for the drug, I have to modify again to find the
target,
for the fluorophores for the thermostat. I think it's very interesting if we can try.
Thank you very much.
Hey, thank you, Andri. Is there any more questions from? Yes, please state your name and institution. Maybe have your Zoom unmute. Okay, thank you.
Thank you. Moderator, my name is Bambang. Unfortunately, I'm not your specific research area, actually. I'm from a research center for fishery. So I just wonder if all of your drugs you have explained also working with fish. I mean, like Dr. Chee, you said about the anti-parasite. This is for the human, right? And
Yeah.
also Dr. Ken, this is on. But I wonder if this also work to the fish. With while we have that kind of drugs, can we apply to the fish also? Because in human there are a lot of drugs already, but in fish there are not a lot of drugs. So if your fish get infected by the parasite, by the bacteria, you don't have any other option. Yeah, you just kill the fish. Otherwise, they will spread the disease to the other. So I just wonder if in the near future or already you have actual information about the application of these drugs to the fish. Please give me some information about that. Thank you very much.
Okay, yeah. Thank you very much for the question.
I'm sorry, I'm not asking about the technical about your research area.
Good question. Yes. Yeah. We have not tried any one of our compounds, especially on aquaculture. But of course, if there are interest to look at some of the compounds that we have basically synthesized, I think we are more than happy to collaborate. Because we do not know whether our compounds are basically active, in aquaculture or not.
Okay.
So if there are some infections of the aquaculture, for example, in fish, that actually may use our compounds, we're happy to collaborate with them.
Okay, cool. Thank you.
I'm not sure about whether Dr. Chee has tried on the compounds on any.
Definitely I haven't tried that, but I think it's an interesting area to go on, especially if the parasites or the bacterial, they are pretty similar or a similar class of parasites that also affecting humans. I think that's potential to test that. For example, some of the antibiotics, like the nitrofuran antibiotics, they are not only human, but they are also used in, for example, animal as well. So if that perhaps we can also test our compound in a similar direction. But we're open for collaborations.
Okay, cool. Thank you.
If you are interested, perhaps we can just perhaps
I think there are many examples
Okay.
that we use both in humans as well as in aquaculture. Nitrofurans and chloramphenicol being the other one. Yeah.
Okay. Because in maybe also the same like in human, we cannot use the antibiotics for treatment the fish also. Actually, if we have used antibiotics, the production will be declining in fish actually. So we don't have a lot of any choice for the treatment of the fish. So if you are allow us to make a collaboration, I think it's good enough. It's good enough. Not anyone in I don't know about the situation in our institution here, but some researcher is scheduled in some research center, so we have difficulties to find a right person to do this kind of research actually. So maybe your help will be useful for this kind of research actually. Thank you very much.
Thank you, Pak. Yeah, that's actually that's something that we are truly expecting, like potential collaboration ideas, and I think Dr. Ken, Dr. Chee will be really happy to be in contact. Can provide you with their contact details, and then have a further discussion for potential projects.
Okay.
So that's
Thank you.
actually a really good question too, like other applications for your project. Yeah. And thank you. Is there any question from the offline participant currently? Now, I actually have a question for both you, Dr. Ken and Dr. Chee. You were both, I think you mentioned in your talks about selectivity. And I guess in the broader context of antibiotic design, the question I'm asking is whether or not you would actually prefer selective antibiotics or broad spectrum ones? Because I guess for selectivity, then you, for example, you have agent selective for gram-positive. Then I guess in just going back to my first year PhD on looking for drugs selective for Clostridium difficile, that would be a little bit more of an interest because of to minimize effects of the drugs with the gut microbiome. But I guess for like antibiotic discovery or drug discovery in general, what would your opinions be on broad spectrum and selective antibiotics?
I think it always has been a long-term discussion, even since I think when I started the project, we always have the issue whether we want to have broad spectrum or selective. I would say that in the market, we can see a lot of broad spectrum antibiotics already available, like ciprofloxacin. It's very selective. It's very broad spectrum against both gram-positive and gram-negative bacterial. And I think it's really depending on the application-wise. For me, if I'm going to develop a drug that is targeting, for example, tuberculosis, I would want something that is pretty much more selective, which means selective only against mycobacterium, but not against other like non-tubercular mycobacterium, because as you say that it could also affect the gut microbiome as well, other beneficial bacteria in the body. But I would say to develop a selective molecule is not easy because some of the bacteria do share similar targets or similar sequences. So it's not as easy to design a selective molecule, I would say. So if you ask me broad spectrum or selective, I would say it depends on the drug molecule itself. If you are able to design selective, why not? But if not, I think broad spectrum could also be something quite useful at the moment. I'm not sure whether...
Yeah, maybe just to add on from what Dr. Chee has mentioned, I think it also depends on who your target people is, because if you are talking which is a little bit more broad spectrum, so that they can use it for a lot more applications. But in terms of research, I think selective ones would probably be preferred to me as personally, I would think, because if you have something which is more selective, it's harder for the bacteria in general to gain resistance because you're not targeting other bacteria at the same time. So the other bacteria would not be resistant to your compound. So it actually prolong the lifespan of the compound. Yeah.
Yeah, I think it's because obviously one of the biggest issues is like the antimicrobial resistance.
Yeah.
Like it's really easy for bacteria, I think particularly gram-negatives, where they, with the horizontal gene transfer, they're transferring antibiotic resistant genes. And like you were saying, if the drug is selective, then the other bacteria might not necessarily be affected. So they're not developing resistance against that particular compound. Okay, so yeah, it's one of those long argument. But yeah, so that's actually interesting. As well as I have another question. Let me see. Yeah, about your I have a question for Dr. Ken. For you were saying about the short peptides, you don't actually have sort of like a delivery system.
Yeah.
So is this because their activity is not affected, or is it because they're actually, being active against bacteria? In terms of, I just wanted to sort of clarify, because I think I've worked a little bit on peptide vaccine, and I guess in terms of their activity, they needed adjuvants and a particular delivery system. So I think with what Dr. Andre asked you previously, whether or not you used the delivery system and you didn't have to, would that be true for all of the short peptides that you've tried? Or is there any particular cases that you found that you actually needed some extra help?
Yeah.
So that's my first question of that one. And the second one would be on, have you actually tried your compounds on clinical isolates with different potentially resistance mechanisms? Thank you.
Yeah. So the first question, probably I'll answer the first question first. For the short peptides, technically we have not come to a stage where we think we are confident enough for the drug delivery. Main reason is because the MIC that we are looking at the current moment is not that attractive for us to move on to the next stage of exploration. Small molecules, at this stage, we have quite good activity, and therefore, at the current moment, we do not need the drug delivery system in vitro. So I'm not talking about in vivo, but in vitro. So at this current moment, but we may need to come to, a different drug delivery system if we go in vivo and we find that, the compounds are not actually reaching the target. Second question on the whether we have tried on the clinical strains. Yes, we have, and we're happy to also share that the compounds, especially the hybrid ones, which are the most active at the moment, they're also active against clinical strains and clinical resistant strains as well.
Yeah. Have you also tried them on, because some of us here are part of the Biofilm Research Collaboration Center, so I think we're also interested in seeing whether your compounds are having activity against biofilm-forming pathogens, and it would be something that could be pursued further, I guess, if you do have evidence that those compounds actually have some activity on biofilm-forming pathogens, to sort of move forward and elucidate, whether how the mechanism of action and things like that to actually, have some anti-biofilm agents.
That's actually a very good suggestion, because we have not actually proceed to biofilm testing as of now. So it's still in the early stage where we can explore a few collaborations, for example. Yeah. And I think that's the opportunity that we are actually seeking, because some of this we do not have the expertise in Monash, Malaysia. So we're happy to collaborate with them.
Thank you very much, Dr. Ken. Okay, we still have time for questions. If then, okay. Yeah, please state your name. Please state your name and institution. Unmute. Thank you.
Okay, my name is Linda Sumarini. I'm from Research Center for Applied Microbiology. I want to ask, maybe I missed some of your slides sometimes, and then I think most of your peptides is synthesized from the known compounds already, or from the commercial one. Then you modify it to become new analogs, right? Or it's also from like nature products, native. I mean native yeah. Then you also modify them, and then when you modify, most of them is like I don't know. I see that it's linear peptide, right? So do you have ever also tried the cyclic peptide? Usually, I think the activity for cyclics somehow is some better rather than the linear peptide, right? So that's why you derived from the linear peptide, right? So
thank you for the question. So yes, the cyclic peptides we have tried, but then we encounter some difficulties in terms of the purification.
Yes.
So we need to probably use HPLCs to kind of purify them. So we are currently at the stage of doing that as well. But for the crude ones, yes, we do see better activities for the cyclic peptides compared to the linear ones. So that's where the modification actually comes in. But in terms of modifying just the linear peptide itself, basically is at the terminal, the C and the N terminal. If needed for stability, for example, if there is a need to stabilize the peptide from degradation, so we could also use the sulfur bridge if needed. But then since it's a short peptides at the current moment, we don't include.
Yeah, because so far I see that a cyclic peptide is like more potent than linear one. That's why most of researchers do this with biosynthesis. So with recombinant things other than this SPSS, yeah, solid phase synthesis. I see because of that, yeah.
Yeah, we could also explore that area as well.
Yeah. Thank you.
Thank you, Dr. Linda. Is there any more questions? I think we still have maybe 1 or 2 minutes. Is there any questions from our online participants? I don't think we have any question. Okay, I think since we don't have any more questions, perhaps for the online webinar, I would like to maybe close the session in terms of our online webinar, because we still have, I guess, for the offline participants, we can still have the opportunity for further discussion with our speakers. And I think one more round of applause for both of our speakers. And I would like to personally say thank you on behalf of the Research Center participants here. Thank you for your time and your expertise and your knowledge. And I'm sure that, of course, you won't mind for us to provide everyone with your contact details for if there's any ideas, potential collaboration ideas, or any sort of other questions and things like that coming from this webinar. And yes, I think we've seen from both of our speakers that there is still opportunities for drug design and using hybrid compounds and small molecules, and there's still potential to develop better drugs to alleviate the drug resistance problem. And we can see that there are certain applications for anti-cancer and anti-infectives that have been presented, and I'm sure there will be other applications that might not be feasible now, but in the future, it could be developed as well for other applications. And with that, again, I'd like to thank you, and thank you everyone who's been kindly, being really good and interactive and asking questions. And thank you also for the organizers for this opportunity. And with that, I would like to close the session, and I will return the session to our MC. Thank you.
Okay, thank you, Dr. Olivia, for leading the session, and thank you for the speakers for their excellent presentations. And as a token of our appreciation, we will give a certificate to Dr. Chee Wei Ang and Dr. Kang Yun Young as a speaker. Okay, this is a certificate. Thank you very much for sharing your valuable knowledge. Okay, ladies and gentlemen, this is the end of our agenda for today. I'm Andhalusi Arini. On behalf of the committee, I would like to say thank you very much to our speakers, moderator, and all participants. Let me end with a short closing poem. Thank you for your kind attention. May knowledge spark your inspiration. Until we meet again, stay in good health. May research continue to grow our wealth. Okay, thank you once again for joining today's webinar. To stay updated with our future webinars, research activities, and publications, feel free to follow and support us through our social media platforms: Facebook, Pusat Riset Mikrobiologi Terapan, Instagram at PRMT_BRIN, and YouTube channel PR Mikrobiologi Terapan BRIN. Wassalamualaikum warahmatullahi wabarakatuh. Have a great day and see you at our next webinar. Thank you very much.
Sorry.
Don't break it. I will put it in.